So the talk that I presented today is about the molecular classification of prostate cancer. In that discussion, I highlighted about the discovery of recurrent gene fusions in prostate cancer. So originally the TMPRSS2 ERG and the ETS family rearrangement was discovered. And I presented our recent work on the discovery of RAF gene fusions, which are identified in a very small percentage, about 1 to 2% of prostate cancer, which is a very rare and a new discovery that was made. And we found this aberration in a very small percentage, about 1 to 2% of prostate cancer. And what is interesting about this gene fusion is the RAF genes are known to be involved in other cancers, particularly melanoma, in which more than about 50 to 70% of the melanoma patients are known to have mutations in the BRAF. And the drugs targeting the mutant BRAF has been already developed, and it is being extensively used for the treatment of the patients. And what we found in our study, the BRAF gene is activated by a mechanism that is not by mutation but by gene rearrangement. So what we found from our early studies that the drug, the PLX4032, that is targeting the mutant BRAF, may not work on the gene that is inactivated by rearrangement. So with this, we identify a new class of aberration involving the BRAF, and also in our study we showed that the oncogenic activity of the rearranged BRAF can be inhibited by the already available drug called the sorafenib or the MEK inhibitors that can be used to treat the patients with the gene rearrangements. So as we know that the drug that is developed for the mutant BRAF may not work for these patients, so we have an entirely new class of drug that can be used to benefit the patients. Also in the talk I highlighted the importance of molecular classification of the prostate cancer based on the gene fusions. Because based on the histopathological observations, it is very difficult to classify the patients and select them for treatment is almost impossible. So the genomic analysis, by using an entirely new approach called the next generation sequencing technology, is emerging as a very alternate and viable approach to understand the actual genetic basis of cancer, and with that we will be able to make important discoveries and which will help patients to classify them based on molecular aberrations and new diagnostic tests, and also the drugs to treat those aberrations can be developed in the near future.